Hello everyone, let's get started. Today we're going to be talking about the motor proteins and their role in intracellular motility. The way we're going to organize the topic today is to first think about the difference between microtubule-based movement inside of a cell and microfilament-based movement inside a cell. We'll take a look at the motor proteins involved, the kinesins and the dyneins, both cytoplasmic and axonemal varieties. We'll talk about the role that the endomembrane system plays in all of this. And we'll look at flagella and cilia and their role in moving cells. There are two very different types of cellular motility or cellular movement. The first and most obvious is the literal movement of a cell through its environment. We know that flagella and cilia are involved in that. We know that microfilaments are involved in amoeboid movement, and we will talk a little bit about that today. We've also mentioned just briefly the fact that microfilaments are involved in the shortening and the lengthening, or what we call contraction and relaxation, of muscle cells or muscle fibers. That can be seen as cellular movement. The cell is literally getting shorter and more compacted during contraction, and then more elongated and relaxed during relaxation. But there's another kind of motility that we're really going to be focused on today, and that's what we call microtubule-based intracellular motility. It's the fact that microtubules are used by a cell as roadways to send materials around inside the cell. In microtubule-based intracellular motility, we're going to see the involvement of a particular class of protein called a motor protein. And these proteins get their names because they are literally like little motors. They will literally move down these microtubule tracts once they are fueled with energy from ATP. So we're going to see this chemical energy in ATP, this potential energy, transformed into this mechanical energy in these motor proteins. A lot of our understanding of microtubule-based motility and motor proteins comes from some experimental work done with axons, the long membrane extensions that emerge from neurons. And we'll talk about that experimental work in just a minute. But what we learned through those experiments was that there are two sort of big groups of motor proteins. There are what we call kinesins, and there are dyneins. Kinesins are responsible for the anterograde movement along microtubules. And anterograde just means forward movement. The dyneins, in comparison, are responsible for retrograde movement along microtubules, or backwards movement. Now, we'll explain in just a minute how you know whether you're going forwards or backwards on a microtubule. There are actually several types of kinesins. There are two subtypes of dyneins. There are what we call cytoplasmic dyneins and axonemal dyneins, and we'll talk about those separately. Now, as I said a minute ago, flagella and cilia are not involved in moving things along microtubule tracts, but are rather involved in moving the cell through its environment. But we talk about them today because flagella and cilia are structurally composed from microtubules. So we will be talking about that type of movement today as well. So microtubules are providing roadways or railroad tracks or pathways or however it is you want to visualize it in your head. They are providing the track within a cell that allows for materials that are inside of vesicles to reach their final destination. These are organelles, these are proteins within a vesicle, these are neurotransmitters. These are all kinds of things that get packed into vesicles and get sent along these tracks to their destination in a cell. Now, we use a term cargo to describe the thing that is being transported by motor proteins. So any vesicle, any organelle that is being carried by a motor protein goes by this sort of generic term cargo. These motor proteins are really fascinating molecules. They have to be one of my personal favorite proteins because they are capable of physical motion. And you're going to see that they almost look cartoonish because they have portions of them that look an awful lot like feet. And those feet are going to essentially walk along the microtubules to get to their destination. They use energy from ATP, as I said a moment ago. The energy in the ATP is what fuels this physical motion. And again, there are two major families of motor proteins, the kinesins and the dyneins. On this slide, you're looking at a table that comes out of a textbook. There's nothing for you to learn from this table, but I just wanted you to look up top here. What they're showing us on the upper part of the table is various motor proteins that are involved in microtubule-based movement. So you can see that there are kinesins here, and there are several of them. And then there are two types of dyneins, cytoplasmic dynein and axonal dynein. These are the motor proteins that are involved with microtubule-based movement. At the bottom of the table are the types of proteins, the motor proteins that are involved in microfilament-based movement. We're going to be talking about these when we talk about muscle physiology and how muscle cells move. So the original work on motor proteins and microtubule-based movement was done using axons. Now, we haven't talked about neurons yet in any detail, but the axon is the longest extension, the longest membrane extension that you find on a neuron. And it is the structure that conveys the sensory information that the neuron picks up along to the next structure. The axon carries that information to the next structure, whatever that happens to be, and it carries it in the form of electrical current. So axons are microscopic structures for most neurons, but there is an axon that you can see with your eyes. It's called the squid giant axon. Now, I will mention this is not the giant squid axon. This is the squid giant axon. So these come out of ordinary squid, but they are literally visible with the naked eye. And that's what you're looking at in the image at the bottom of the slide. You can see a person's hand here, and there's a dish. The person's holding a dropper and is manipulating this thing that looks like a white thread across the surface of the dish. This is an axon that comes out of a squid. It's called a giant axon. and it makes a wonderful model to study motor proteins. Essentially, what happens is that substances get generated up in the cell body of a neuron, including things like neurotransmitters. And then those substances get packaged into vesicles, as we've talked about. And the vesicles then have to be sent all the way down the axon to the terminal portion of the axon where it's going to synapse with the next structure. It's either going to synapse or communicate with another neuron at that point, or with a muscle or a gland. So imagine the work involved with getting a vesicle from the neuron cell body all the way down the axon to that terminal region where the synapse is. Those vesicles have to be physically transported, and they're transported along microtubules. Now, in the squid giant axon, we're talking about an axon length of about 3 to 4 centimeters. From a cellular perspective, that is just an amazingly long distance. It has to be done, though, and it has to be done fast. It has to be done in real time. So this is a process that got called fast axonal transport. The substances that are packaged in the neuron cell body in the squid giant axon are transported down the length of the axon at 400 millimeters per day. If you think about it, that's pretty impressive. So it turns out that fast axonal transport happens not just in squid, but in all axons. It's also occurring in flagella, and in both of those structures, it's helping to move cargo to its destination. On this slide, you're looking at a very basic drawing of a neuron. So this is the cell body of the neuron. Here's the nucleus. Here is the axon. And remember, this is just what we call an extension of the membrane, as are these little structures here called dendrites. Again, we're going to be talking about neurons in detail in a couple of weeks. But for now, understand that information is received by the neuron at the dendrites. It's then processed and sent along the length of the axon to the next structure, whatever that structure happens to be. And we said that might be another neuron, it might be a muscle, it might be a gland. At the junction between this neuron and the next structure, there's what we refer to as a synapse. And depending on the type of synapse, that sometimes involves neurotransmitters being released from this neuron to the next structure. Those neurotransmitters are packaged into vesicles. They're created up here in the cell body, packaged into vesicles, and then sent down to the synapse. Now these green linear structures that you're seeing here, these represent the microtubule tracks that we've been talking about. And the little dots that you can see, the little red dots, represent motor proteins. These motor proteins are traveling back and forth along these tracks from the nerve cell body, where the neurotransmitters are made, to the synapse, where they're used. So motor proteins are key to this. The motor proteins are the workhorses of fast axonal transport. And like I said earlier, they are literally capable of walking down the microtubule pathway. And they need energy in order to do that. Now, we said that the kinesin variety of motor proteins performs what we call anterograde transport or forward transport. Now, how do you define forward transport? We define it as heading towards the plus end of a microtubule. Remember, the microtubule has directionality. It has a plus end and a minus end. Kinesin is going to travel in an anterograde fashion. It's going to travel toward the plus end. Dynein does the opposite. Dynein motor proteins follow retrograde transport or backwards transport, so they're going to travel toward the minus end of microtubules. Now, on this slide, we're looking at a drawing of a kinesin motor protein and a dynein motor protein. Both of them are carrying cargo, and they are situated on top of a microtubule. So here's our microtubule, and you've seen these types of drawings before. You can see the individual heterodimers inside the microtubule. The gold ball and the yellow ball together represent one heterodimer. Here's the plus end of the microtubule. That would be where new tubulin could be added. Here's the minus end. That could be where tubulin could be removed. Remember, microtubules are constantly treadmilling. And here is a kinesin. I told you this thing looks cartoonish, right? You could see that it has what almost looks like a body and two arms at the top and two little feet at the bottom. And these really do work the way feet work. These will literally move along the length of the microtubule using the energy from ATP hydrolysis. And because it's a kinesin, it's going to be moving anterograde. It's going to be moving towards the plus end. Now, dyneins look similar, but a little different in their structure. They're different proteins, but they too have what looks like feet. And their feet are going to walk this way, retrograde, towards the minus end of the microtubule. So let's talk first about kinesin-based movement along microtubules. So kinesins structurally are composed of three things. They're composed of what are called globular heads. Now, this is what I refer to as feet. They're composed of heavy chains and light chains. Now, this chain terminology, this has been around for a long time. We use heavy chain, light chain terminology to describe regions of proteins. So in a kinesin, remember what I referred to as sort of the body, that's heavy chain. What I would call the arms, that's light chain. And what I would call feet, those are globular heads because they are globular in their shape. Now, on this diagram, you can see different varieties of kinesins. Remember, there are multiple types of kinesin proteins. They're all a little different. They're different sizes. The length of the heavy chain really varies. The number of globular heads varies. But they're all similar in that they all have heavy chain, light chain, and globular heads. Now, when we look at those globular heads, we can talk about some regions within those heads that we need to be familiar with in order to understand how these proteins are capable of movement. These heads have what's called a catalytic core inside of them. They have what's referred to as a neck linker region. And importantly, they have an ATP binding site. So those structures within the globular heads, within what I'm calling the feet, those structures are going to be important to our understanding of how this protein moves. The light chain regions of a kinesin are the regions that actually contact the cargo, these arm regions as I call them. They're actually what is holding on to the cargo, whatever that happens to be. And the heavy chain is sort of what connects the heads to the light chains. The heavy chain is sort of that central region.